All right, guys, good morning, good afternoon, whenever, or good evening, whenever you guys are listening to these tapes, these recordings. So we talked about chapter 5 so far, and now the remaining of chapter 5, we need to go over that. And again, everything on the remaining of chapter 5 is important. I would like you to know, and then whatever we say in this chapter, it is needed for the rest of semester, as I said. So this is a nice chapter for the basic foundations of the rest of the semester. Proteins include a diversity of structures resulting in a wide range of functions. Proteins account for more than 50% of the mass of most of the cells. Okay, so a lot of proteins are found in the cells. That's the most abundant macromolecules, if you would, in a cell, because as I talked about it before, the cytoplasm of the cell, which is the ground of the cell, the medium of the cell, that's the word I was looking for. For the medium of the cell, it is made up of protein and water. Some proteins speed up chemical reactions, okay? Yes, like enzymes, we already talked about that. And other proteins function include defense, storage as an energy. You remember 1 gram of protein gives you 4 kilocalorie. Okay. Transports, we'll talk about that. Cellular communication, movement, or structural support. All of that, all of these functions, we will talk about them throughout the semester. Not necessarily, we briefly talk about it here now, but for the rest of semester, we will talk about it. Excuse me. I don't know why these things don't work. I want to go to the next slide. I guess I have to come here, click, and now, okay. I'm not going to read these up to you guys, but I would like you to read them, to know them, look at them, the defense, the enzyme proteins, and storage, and so on and so forth, transport. Again, we'll come back, but it's good that you have a general idea as far as what's going on with these protein molecules. Remember, guys, whatever you're learning in this class, you do need it for the rest of your career when you go into biological field. You're going to become a doctor, you do need these. You know, for the standardized exams, you have to take MCAT, USMLE, USMLE2, DAT, the board certified exam for DA, for becoming a dentist, pharmacist. You do need this information. So just do not let, you want to become a teacher, you want to become a researcher, whatever you want to become in biological field, you do need this information, all of this. And then that's why I'm asking you, make sure you know them. Here is the rest of them, hormonal proteins and receptor proteins. Oh, they offer courses in major universities on receptor proteins. And we'll talk about receptors for a good 2 chapters. Cell membrane, and then we'll talk about receptors as well. Then contractile and motor proteins and structural proteins, they're all important. The structural proteins, we talk about it briefly, not much. When you get to zoology, we'll talk about it a little bit more. Okay, contract, you know, it is great. You should watch these on your own. Please watch them on your own. I'm not going to go over them because you do need them for the rest of semester. You do need these ideas, whatever is being said on these animations, you do need them. Again, there's defense, enzymes, gene regulatory proteins. That is very important. We spend almost, toward the end, we spend a good amount of time on things like this. So make sure you know them. Hormonal proteins, we don't spend that much time quickly. We spend a lot of time later on gene regulatory proteins. Maybe we should watch this one together. Let's watch it together. Just this one, because how come it doesn't come out? Oh, I gotta go to, oh, oh, here we go. Interesting. Hey, maybe I should pick that up. We used to be able to have them, but I don't know. Okay. Well, you guys watch them on your own, please. I thought maybe it has something that we need to talk about it. Okay. Hormonal. I don't know why I don't get my mouse. I used to get mouse. Sensory proteins, storage proteins, structural proteins, and transport proteins. They all have something to mention, and I'm hoping you would watch them. And there could be some exam questions from them. So watch them. Not necessarily, I'm not going over right now because I strongly believe they're all very good. You know, if you have any question, holler at me, send me a note. If you want to have a Zoom session, we talk about all of this. Okay, so proteins are made up of what? Amino acids. Amino acids are monomer. So in human, we have 20 different amino acids, and you see them, you're going to get to know them very well. I don't expect you to memorize them, but you will get to know them. And then the amino acids, so as we go on, I want to say a lot of stuff, but I'm sure the PowerPoints come up. It will say what I want to say. But amino acids are made up of 1, 2, 3, 4, 5 component main parts. So let's start from left to right. The very first part is your amino group. You already know that. The second part is your central carbon. They call it central or alpha carbon. I like to call it central carbon. Okay, then you have your acid group, number 3, the hydrogen, number 4, and the R group, number 5. So 5 components, and all amino acids they have these 5 parts. And what makes amino acid different is the R group sides chain. Okay, that what makes amino acids different right here. So we have 20 amino acids. All 20 amino acids have part 1, 2, 3, 4. It's a part 5 that those 20 amino acids are different. Okay, so and all proteins are made up of this. It is just like in English language, how many alphabet do you have? You have 26, 24, whatever it is. And then how many words do we have in English language? Let's say 500, 1,000, 500,000. One linguistic was telling me that. I could be wrong. I don't know. I never read it anywhere. This gentleman, he used to teach at Delta College. He told me that. So let's say 500,000 words in English language. And then how many books do we have? Can you give in English? It's unlimited. Okay, so that's how it is with protein molecules. We have 20 letters, but the amount of protein molecules is unlimited. It's just infinite. Okay, so let's move on. Here there are 20 amino acids. I would like you to know them by name. When you take biochemistry, you have to memorize the structure of each one. The R group, of course. You have to memorize the R group of each one of them. In this diagram, they draw what they have in common in blue boxes right here for you guys, and the R group in yellow boxes. And then I also would like you to know for this class that which amino acids, and I'm gonna go over them and read them to you guys so you know the pronunciation, which amino acids are non-polar, you should know that. Which amino acids are polar, and you know the definition of polar and non-polar. Polar molecules are like water. One end it can be positively charged, the other end is negatively charged. And then you have 2 amino acids that are they're acidic, okay? And then 2 amino acids that they're basic, okay? They're the pH, if you would. That's what I mean by acidic and basic. The pH if they're in a solution with a pH, the pH is between 7 to 10, 7 to 7 to 14, sorry about that. And then here is 0 to 7. You know what acid and base are. Okay, let's go over. The nonpolar amino acid, they're hydrophobic. You know that. They do not like water, hydrophobic. They dislike water. Or glycine, the simplest of all amino acids, glycine. And then you should know the abbreviations. GLY is for glycine, or G. I like the GLY, but anyhow, make sure you know them. Alanine, ALA, valine, leucine, isoleucine. Okay, those 5 are non-polar. Oh, I'm sorry, there is more. Methionine, phenylalanine, tryptophan, and proline. Proline is a little bit, it's forming a ring, so you should know these things. Proline is the only amino acid, not each one of them, some of them has a only, okay, so glycine is the simplest, right? Proline is the only amino acid. It forms a ring, you guys can see the ring, with the commonality parts, with the common structure. So the R group is coming back to the nitrogen, to amino group right here, and forming a ring. So study them and study the structures a little bit. It says, well, you don't have to know the structures, but when you go to biochemistry later on, you have to know the structures. So it's nice that you get familiar with them. Okay, and then the polar ones, the polar amino acids are serine, 3-ionine. 3-ionine is an amino acid that has, I'm sorry, no, nothing, don't worry about that. Serine, 3-ionine, cysteine. Cysteine is one of the amino acids, there are 2 of them, that have a sulfur group, okay? So look at the sulfur, it has a sulfur. And then tyrosine, asparagine, and glutamine. Those are the polar amino acids, all right? They are polar, they are hydrophilic. The electrically charged side chains are hydrophilic. Okay, the negatively charged, I should move this up a little bit. So I don't know if you guys can see that or not. The acidic amino acids are aspartic acid and glutamic acid. And the basic amino acids are lysine, arginine, histidine. Those are the those are the 3 basic amino acids. There is one more amino acid, I skipped it, I'm sorry about that. I skipped it, which is, has sulfur, and that is methionine, right here. Pointing right here. That's methionine. Methionine has a sulfur group as well. And cysteine, of course, it has also a sulfur, sulfur hydryl. We talked about that. The sulfur in methionine is not sulfur hydryl. The sulfur in cysteine, it is a sulfur hydryl. Okay, here are the bigger pictures of them, the non-polar ones. Again, the only two of them you should know. Well, all of them you should know, but characteristics-wise, glycine is the simplest one, and proline forms a ring. These guys have ring also, right? But I'm not talking about that type of ring. I'm talking about the ring. The R group is forming a ring with the ammonia group. Okay, these are the polar ones: serine, 3-ionine, cysteine with the sulfur. Oh, I forgot to mention. How come I forget? The methionine. It's also very important amino acid. Methionine, when we talk about protein synthesis later on, methionine is always the first amino acid. Now it gets chopped off, or it would not stay in the structure. That is, something we will discuss. But methionine is the first amino acids in protein synthesis. And then, of course, cysteine has also sulfur. So make sure you study those. Acidic amino acids are aspartic acid, glutamic acid, and lysine, arginine, and histidine. They are the basic ones. These two amino acids, aspartic acid and glutamic acid, look at them. They are right here, okay? And they are the polar side chains. They are the polar amino acids, okay? Again, I'm not going to go into much of the biochemistry of these things. When you take biochemistry, you will learn them. So asparagine and glutamine, they can be found in an acidic format, which is aspartic acid and glutamic acid. Okay, let's move. The polypeptide, let's see. Okay. The polypeptide amino acids polymers, peptide bond. So I should talk about peptide bond. When you have two amino acids come together like this, they form a bond, a covalent bond between the two, these two right here, and that is called a peptide bond. Remember, we had ester linkage, we had a glycosidic linkage, and now this is a peptide bond. It's not a linkage, it's a peptide bond, and you will see it in a minute. A polypeptide is a polymer of amino acids. We have dipeptide. Yeah, you will hear that definitely in your life later on. You have a tripeptide, 3 amino acids, but you never ever heard of triglyceride or not. I'm sorry, not triglyceride, trisaccharide. I never threw that term at you. That term does not exist. The term trisaccharide, okay? But the term tripeptide exists, okay? And that is 3 amino acids. Oligopeptide, oligo, you know, it means a few. So you have anywhere between 4 to 10, and polypeptides is 11 to 50, and protein is 50 to infinity. Make sure you know that nomenclature a little bit. It comes in later on in your life. Very important. Now, all protein molecules, you will see some pictures, the diagrams I'll show it to you here in a minute. That one end of the protein molecule is carboxyl end, or C terminus, they call it. So the rest of in October, November, December, when I say C terminus, then you should know what I mean. You should have studied this. C terminus, it means, that's the only time we use it, C terminus. It means one end of the protein molecule has the carboxyl end, the acid end, excuse me. And the other end has N terminus. The N terminus, it is the, excuse me. The N terminus is the amino group. That's how, imagine, this is a protein molecule right here. Can you guys see that? One end right here has the C terminus, the acid end, and the other end right here that has the N terminus. And you will see it, hang on. But it came up in this PowerPoint, and then, well, oh, come on. Oh, I got my pointer now. I'll be darned. Okay, right here, 2 amino acids are being combined together. This is the peptide bond, covalent bond, right here. So 1 amino acid, this is 1 amino acid, is going to be combined to that, these 2 dipeptide to become tripeptide. So this, the H, the hydrogen of that amino group is combined with the OH, hydroxyl group of that acid group of that amino acid, of this amino acid, okay? The H of this amino acid is combining with the OH of that amino acid, gives you water. And then there's a covalent bond between them, and that covalent bond is called a peptide bond, right there. So in this molecule, in this tripeptide, how many covalent bonds do we have? 2. How many water molecule combining these 3 amino acids? These 3 amino acids, combining them together, how many water molecule did you create? 2. Yeah, 3 amino acids, 2 water molecules, 3 amino acids, 2 covalent bond peptide bond. Yeah. Okay. I hope I'm making some sense. Think about it. Go over it. I'm not saying nasty stuff. Okay. Animation, protein structures, introduction. Sure, let's, I wanted to see that one.
Most proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure.
Okay, that's the next thing. 4 levels of structures, 4 levels of protein structures. The primary one, structure of a protein is unique, sequence of amino acids. Secondary structure found in most proteins consists of coils and folds and polypeptide chains. Tertiary structure is determined by interactions among various side chains of our group. Quaternary structure results when a protein consists of multiple polypeptide chains. Okay, here's a primary structures. For example, in your insulin, if you look at the amino acids right here, is one end, this end of their all chain is amino end, and at the end, that end, if I may, right here is the carboxyl end. My marker doesn't write on it. So that's what I meant by the end. That's A terminus, right? A terminus, and this is C terminus. Okay, and then the rest are, they wrote you the three letters of amino acids. And remember, between this carbon and that nitrogen, this is a peptide bond. This is a peptide bond. The next one is a peptide bond. So I did mark for you guys, this is a primary structure. Then what you have, the secondary structures, you have 2 different secondary structures. You have alpha and you have beta. Again, you saw them in glucose molecules when they are alpha and beta, you also saw them. Sit down and study them and know the differences. Okay, the alpha helix, it means the protein molecule is being, all of these are amino acids. Can you guys see this? Okay, so all of the the way it is, the protein, the amino acids are being twisted like that. Okay, that's an alpha, if you would. This is the alpha structure. Of course, there is a hydrogen bond between the amino acids of one amino acids and the next amino acid, and so on and so forth. So this would be an example of an alpha helix secondary structure. Okay, so right here is a hydrogen bond between that amino acids and then that amino acids. Okay, so you saw the hydrogen bond. Then if you have the beta, the beta proteins, they're pleated. They're like, they call them, that's the term they use in all textbooks, pleated. They're like this. Okay, can you guys see this? So they are pleated structure. They're not twisted, they're pleated. This is a beta structure. Now all of the amino acids, I did draw them for you. And the way they are, again, hydrogen bonds come into action. So as you can see, these are all pleated structures. And then of course they form a hydrogen bonds with the next chain of amino acids combined together. And of course, between them, there is a hydrogen bond. The examples, I gave you a couple of examples of actin in muscle cells. Okay, usually it's alpha structures in amino acids, and then in muscle cells, and then you have tertiary structures when you have these combination. Well, it's hard for me. If you have combination of these in a protein molecule, you have the alpha helix. Okay, you guys follow me what I'm talking about. It's combined, portion of it is like that, and portion of it is, they're all together. I should have brought you a tape and tape them up together. If I would have taped this together, that would be a tertiary structure. Okay, this is a tertiary structure. An example of the tertiary structure is a myoglobin. Myoglobin is found in your muscle cells. Okay, then you have quaternary structures. So imagine what I had right here, the helix, the alpha helix, and then you have a beta one. You have another protein molecules right here separately. You combine all of them together. So that would be a quaternary structure. The best example of quaternary structure I have is in hemoglobin in your blood, which is made up of 4 subunits. Okay, 2 alpha and 2 beta. When you study hemoglobin structures, you will know them. And right now in this course, we are not studying them. We are not taking hematology or biochemistry. But no, hemoglobin is a quaternary structure. Okay, and then it's a combination of several units. So you have a protein molecule here, a protein molecule here, combined together, and that would be a quaternary structure. Primary protein structures, I think we talked about this. I don't know why it was coming back again. My mistake, I guess. The coils and folds of secondary structure results from hydrogen bonds between repeating constant of the polypeptide chain backbone, typical string structure or coil called alpha helix, and the folded structure called beta pleated sheets. Okay, we talked about all of this. Again, secondary structure proteins. Well, let's watch it since my clicker is working.
In most proteins, parts of the polypeptide chain are coiled or folded,
right,
forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself, or where 2 regions of the chain lie parallel to one another.
Right.
Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself.
Okay, the hydrogen bond, we talked about it earlier, so I'm not going to spend too much time on hydrogen bonds and move on. And idealized alpha helix, let's move on. I don't want to take up an idealized alpha helix, no side chain. You guys can watch these if you want. I'm not too crazy about these videos. The previous ones, the structure, the functions of proteins, yes, you should watch those. Tertiary protein structures, let's watch this one quickly.
Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall 3-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups.
Okay, quickly, he brought it up. I don't know in the PowerPoints it's going to come up or not. Disulfide bond. So you have a cysteine here and a cysteine here. Do you guys follow what I'm talking about? So the cysteine from that chain, that sequence of amino acids, and the cysteine from here, that sequence of amino acids, they form a bond, which is called, it's a covalent bond, which is called disulfide bond. We do have that. I will talk about that for the rest of semester. When we get to zoology, you do need to know that disulfide bond. Disulfide bond comes from cysteine, and a cysteine, the amino acids has sulfur group, and they form a covalent bond. It's a very strong, you know, as we talked about it, covalent bond is the strongest bond of the ones that we studied, and it's very, so the separation of them is very tough. So, for example, if you have curly hair, you naturally curly hair, you have a lot of disulfide bond. If you have smooth, flat hair, you do not have that disulfide bond, much of it in your hair. When, for example, those people who have hair that are curly and they want to make them straight, they iron it. So they break down these disulfide bonds. Okay, that's the only way. The people who have straight hair and they want to make them curly, they add chemicals to it that has sulfur, and it makes, and of course they iron it a little bit, curl it a little bit, adding the chemical that has sulfur, so it becomes curly. Okay, I hope I'm making some sense. Okay, tertiary structure, the overall shape of a polypeptide results from interaction between R group rather than interaction between their backbone. These interactions include hydrogen bonds. We talked about that. I talked about it. The video talked about it. The disulfide bond reinforce the protein structures as well. Here we go. So they are talking about these. You guys, please study these later on. We do need them. And when the time comes. Coordinate structure, sure. Let's watch it, see what they have to say.
Some proteins consist of 2 or more polypeptide chains.
That's what I talked about.
The fourth level of protein structure, quaternary structure, results from the combination of 2 or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of 2 kinds of polypeptide chains. 2 of each, a total of 4 chains, make up each hemoglobin molecule.
Okay, I talked about that for you guys. Beta chain, 2 beta chains, and in between right here, you don't want to worry about iron. There's iron here, iron here. So each hemoglobin molecule can carry 4 oxygen molecules for you guys in your blood. Okay, coordinate structure is also when 2 or more polypeptide chains form one macromolecule. Collagen is a fibrous, yes. The most common protein molecules on planet Earth is collagen. We talked about that. So collagen is the most common macromolecule on planet Earth. And then hemoglobin is a globular protein, like a glob. That's what it is. Protein consists of 4 polypeptides, 2 alpha and 2 beta. Yes, okay, your textbook mentioned it, so you should know that. Collagen is a fibrous, tough protein molecule. As you can see in here, there is 1 alpha helix here, 1 alpha helix here, 1 alpha helix here. So that's wrapped around, very tough. You have it in your skin, you have it in your, oh boy, all over. As I said, the most common, the most abundant protein molecule on planet Earth. Okay, again, this is a coordinate structure, alpha unit. We talked about, oh, sorry, I lost my place. Okay, so normal sickle cell, he's talking about that now. Let's leave it for when we get, I'm not gonna ask anything about sickle cell in your next exam. We'll leave it, but study it a little bit. It's just one protein molecule right here, quickly, right here is being different. And because of that one amino acid, not protein molecule, because of that one amino acid right here, then yeah, the sickle cell is formed. We'll talk about it later. Normal protein, denaturation, when protein molecules, I don't know if I threw that term at you before or not, but denaturation is when the normal protein molecules lose their shape. This is the normal shape of a protein molecule in a cell that functions and does perform all of these duties, but when its shape changes, it cannot do those duties anymore. And what causes denaturation? pH. If you put strong pH, strong acid, or strong base, if you put them through this protein molecule, they cause denaturation. Another one is heat. When you fry an egg, it's not same protein molecules as when you fry them. Another one that your textbook did not mention, I would like to mention it, is the physical or mechanical forces. So if I beat that protein molecule, mix it up very well, it's going to change its shape. It's not going to be safe. So that is something your textbook did not mention it. I like to mention it. So, and then if in some cases, a protein molecule can go back to its normal shape in a biological setting, it is called renaturation. Protein foldings in the cell, very important. We will talk about it in next chapter, but you should study, you should know all of whatever we're talking about it in this chapter. It is hard to predict a protein structures from its primary structures. Most proteins probably go through several stages of their way to, become stables. They go, in the plasmic reticulum, makeup will, you will study these things. In the plasmic reticulum, makeup, and then they go to Golgi apparatus, fold them. That's what he's talking about. Go through several stages of their way into become stable. Chaperones, so we have protein molecules that they are monitoring the folding, becoming straight or whatever you have. Diseases such as Alzheimer's, Parkinson's, and mad cow disease are associated with misfolding of the protein molecules. So here they are talking about a little bit of chaperone and folding and what happens with the protein molecules. So the protein molecules that needs to be folded become like this. It goes through the chaperone. This is also a protein molecule. This is the chaperone right here are protein molecules. It goes through and then right here it comes out. And then, okay, done with protein molecules. And then the next one, nucleic acids. I think protein molecules took most of the time for this chapter, very important. But of course, nucleic acids are also important, but we'll talk about them in greater detail later on when we talk about genetics. Then protein molecules and the nucleic acids come together. So let's go from whatever you have to know in this chapter. Nucleic acid stores, transmit, and help express hereditary information. The amino acid sequence of a polypeptide is programmed by a unit of inheritance called gene. Yeah, you probably know that, you already know that. It's the gene that tells what amino acids are going to come out. It's just like construction workers look at the blueprint of something and then the genes come out. Okay, I hope I'm making, the construction will just need some gene color. The building comes out. So the blueprint that the construction workers look at, the blueprint is gene, your DNA. And the building you see at the end is the protein molecule. I hope that analogy helps you. So, and of course the blueprints, the engineers sat down and draw it and, analyze it, the forces, can hold this building hold or not, or the bridge, whatever you are making. Okay, genes consist of DNA, nucleic acids made up of monomers called nucleotide. Yes, definitely. And there are two types of nucleic acid, the deoxyribose nucleic acid. Look at the reason they call it DNA. Right here, D, it means D right here, nucleic, N right here, and acid, A right here. Okay, the deoxy. Deoxy means without O2, oxygen, I should say oxygen. But anyhow, ribonucleic acid are, oh, my here, he doesn't like it, but there are N, and acid right here. So RNA is ribonucleic acid. This one has with O2. Okay, so this is the central dogma of, in biology, you have DNA, the blueprint right here, and then mRNA synthesized. mRNA comes from the nucleus. This is the nucleus. It comes to the cytoplasm, goes through ribosomes, and amino acids are being synthesized right on the bottom. Put together. Amino acids are being put together, and then you have the protein structures. Who decides what amino acids are going to be together? Right here, the DNA. Okay, components of nucleic acid. Nucleic acids are polymers called polynucleotides, and each polynucleotide is made up of monomers called nucleotide. Okay, so each nucleotide consists of nitrogenous base, which we'll study in DNA and RNA. They're different. There are 4 of them. Those nitrogenous bases, there are 4 of them, both in DNA and RNA. And we'll compare DNA and RNA here in a minute. Hang on. A pentose sugar molecule, 5-sided. That pentose sugar molecule is 5-sided and more, and 1 more phosphate. So we'll talk about it. The portion of the nucleotide without the phosphate group is called a nucleoside. Nucleoside is nitrogenous base plus sugar. That's it. Okay. There are 2 families of nitrogenous bases. I'd like to pictures come up in a minute. Hang on. Pyrimidines, yes. Pyrimidines, you have 3 pyrimidines and there are 2 purines. So here is 1, 2, 3 pyrimidines, and then 2 purines are adenine and guanine. The 3 pyrimidines are cytosine, thymine, and uracil. The way I memorize this is TUC. TUC is the pyrimidine, and Aggies, you know, those of you going to UC Davis, Aggies are purines, okay, adenine and guanine. You will see purines is a smaller, shorter word. Look at this. This is a longer word right here. Purines have 2 rings, and pyrimidines, it's a bigger word. They have 1 ring, and you will see it. In DNA, the sugar deoxyribose is, and in RNA, the sugar is ribose. Okay, so here are your nitrogenous bases. Cytosine, do not confuse it with cysteine. Cytosine is a nitrogenous base. Cysteine is an amino acid. Thymine, right here, and uracil. Those are the 3, and I said 1 ring. You remember that? Pyrimidine is a longer word. That's what I meant. They have 1 ring. And then purines, shorter word, compare, it has 2 rings. And adenine, guanine, and TUC. You remember that? TUC, thymine, cytosine, and uracil. Okay, so if you have DNA, 1 strand of DNA right here, and these are your phosphate, sugar, nitrogenous base. So the legs of the ladder, if you would, I brought you a ladder right here. So this would be phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. You guys see that? I hope. So let's go over it. I messed up a little bit. Phosphate, sugar, phosphate, sugar, phosphate, sugar. So the steps of the ladder are attached to the sugar molecules. Okay, right here is the sugar molecule. That's where the steps of the ladder are attached to. So they, on this diagram right here, they just draw for you guys one of the legs of the ladder, one of the, half of it. So these are nitrogenous base, either it's a 2-ring, purine, pyrimidine, so on and so forth. Okay. What is a nucleotide? A nucleotide, we talked about it, it's a nucleoside. The nucleoside is a combination of a nitrogenous base plus the sugar molecule. Okay. And then a nucleotide is phosphate is attached to it too. So make sure you know these terminologies for exam purposes. And nucleotide is combination of all three of them, and nucleoside is combination of these two only. And these are your nitrogenous bases. Nitrogenous bases are broken into two parts, pyrimidines and purines. Pyrimidines have one ring and is tuck, and purines are ag, okay, and they have two rings. Okay, another one, the difference, I wish this was shown up a little bit on the top, the difference between deoxyribose, DNA, and ribose, sugar molecule. Let's count the carbons, carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. So they only have five sugar molecules, 5 carbons, I'm sorry, and it's 5-sided. Pentose is 5-sided. It's a 5-sided, it's a pentagon. Pentose is it's a pentagon. It's a sugar molecule, okay? And it has 5, glucose has 6, fructose has 6. Fructose has 5 sides, right? Fructose have 5 sides, but it has 6 carbon. Right here, pentose has 5 carbons and is 5-sided. So the difference between deoxyribose and ribose is right here. This sugar number 2, carbon number 2. Come on, carbon number 2. Carbon number 2, it does not have the oxygen. Here, carbon number 2, it has, right here, it has the oxygen. That's it. That's the difference between DNA and RNA. Well, there are other differences too, between deoxyribose and ribose. Deoxy, as I said, it means without. Here we go, they're showing you the differences between nucleoside, nucleotide, nitrogenous base. So make sure you get those terminologies straight in your head. And then the differences between nitrogenous bases and purines. T is in DNA, and U, yes, RNA is only in uracil is only in RNA, and DNA does not have uracil. Here's the differences. Again, I talked about it. And here is DNA structure, animation. Let's see what it has to say.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide.
Yeah, we'll talk about that later on.
RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The two polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
Okay, that was great. DNA double helix, I'm gonna, bioinformatics uses computer softwares and use other computational fields resulting for sequencing many genomes, analyzing large sets of genes, or even comparing whole genomes of different species called genomics. A similar analysis of large sets of proteins, including their sequences, called proteomics. So in summary, nucleic acid polymer of nucleotides, also called polynucleotide DNA. Nucleotides is a monomer, and then nucleotides are nitrogenous base plus pentose sugar plus phosphate. Okay, and then nucleoside is nitrogenous base plus just a pentose sugar. Nitrogenous base are pyrimidine, which are cytosine, the TUC, and or cut, CUT, here I use it cut. And then the ag is the nitrogenous base of purine. Okay, going back to the beginning of the cell, of the structures we talk about, if you have a cell, a plant cell, most plant cells are rectangular, like this, or like that. Okay, so the plant cells are rectangular, and then the very outer layer of the cell is cell wall made up of cellulose, right? And then the next layer to it, yellow in this diagram, it is your cell membrane made up of phospholipid. Then you have nucleus in here, which is made up of your nitrogenous bases, your nucleus, nucleic acids. And then inside of the cell, in the cytoplasm of the cell, you do have, you know, deposits of starch, fats, and so on and so forth. And then, of course, the cytoplasm inside of the cell, all of the cytoplasm is made up of, protein and water. So if I ask a question, what building blocks makes up what, then you should know that the building blocks are these guys right here, okay? And then the structures are cell wall, as you know, cell wall, and then cytoplasm. Those are like a wall is made up of what? Made up of cellulose. Window, sorry. A window, what is a window made up of? What is the bottom of? What is the wood? The floor of the house is made up of the wood. I hope I'm making some sense, making an analogy at the same time I'm talking about these things. Plants, animal cells are usually round, okay? So they do not have cell wall, they have cell membrane. Okay, cell membrane is made up of phospholipid. We talked about that. And then inside of the cell, you have nucleus, of course, right here, made up of your nucleic acid. And then you have deposits of glycogen or fat. And then, of course, you have cytoskeleton. Plant cells usually do not have cytoskeleton. And those cytoskeletons hold up the cell. And that is what. Okay, that's the end of the material for this exam, guys. Make sure you study them. Make sure you know them. We need the information for rest of semester for this exam. All right, I'm gonna cut it short. If there's any question, we'll discuss it in our Zoom meetings.